Okay, hello. We are going to do a revision video on monoclonal antibody therapy. So there are 3 revision questions. Pause the video and complete those questions, and then we shall go through answers. Okay, so the 2 types of monoclonal antibody therapy. The first, direct, and you should think of this as a blocking therapy. So let's take a look at what we mean by that. Let's say we've got some cells and the V-shaped structure on their surface is a receptor. That might be a receptor for a growth factor protein. And under healthy circumstances, that growth factor may bind and just help the cells grow. But in a disease state, that growth factor may bind and actually cause the cell to divide uncontrollably to form cancer cells. And we know that cancer is uncontrolled cell division and can lead to the formation of a tumour. So the way direct monoclonal antibody therapy can help in this situation or treat this problem would be to bind to the receptor. So the antigen binding site on the antibody will be specific for that growth factor receptor. And if it binds to that receptor, the growth factor can no longer bind, and so there will be no more uncontrolled mitosis. Alternatively, the monoclonal antibody could have an antigen binding site that's specific for the growth factor. And so by binding to the growth factor protein, it can prevent it binding to the receptor, and again, it prevents that uncontrolled mitosis. So let's look at the other type. Indirect, and you should think of this as drug delivery. So let's look at an example. We have some cells, and the 3 cells on the right are diseased cells, and they will have a specific antigen. What we can do is produce monoclonal antibodies that have an antigen binding site that is specific for the antigen on disease cells, and to those antibodies you can attach a drug. This might be a chemotherapy or radiotherapy drug. Those antibodies will specifically bind to the antigen and deliver that drug directly to those cells and destroy them. So they're the 2 types of monoclonal antibody therapy you need to know about. Let's answer question 2 and 3 now then. So the advantages. So because they're specific, because they're just binding to the disease cells or the molecules that cause disease, it means you don't require such a big dose, so small dose is only used. This means you have reduced side effects, and because you're using less, they're potentially cheaper. So let's look at ethical issues. So some people are opposed to use of animals in medical research. Mice are used in the production of monoclonal antibodies. It's important that the animals are treated ethically and suffering is reduced as much as possible. Ideally, no suffering to those animals. Secondly, we have to test the drugs before they're given to patients, and humans have to be involved in that testing. And so those participants in those trials put themselves at risk of side effects, but that's something that has to be balanced with eventually being able to use those drugs. And finally, monoclonal antibodies are quite a new therapy, so even though we do trials, there still may be unknown side effects that affect certain people or appear after long-term use. And so patients have to be aware of that, and they have to agree to take those drugs and agree that they are putting themselves at potential risk. Okay, so that is monoclonal antibody therapy.